Hello everyone, welcome to our ASCP review session. Today we'll be going through a series of multiple choice questions that cover key concepts you need to know for the exam. For each question, we will analyze the option, identify the correct answer, and understand why is correct and why the other choices are incorrect. Let's begin. Our first question is, which of the following is characteristic of primary granules? A. Coated with a phospholipid membrane. B. Called eosinophilic or specific granules. C. Contain myelopulsitis and lactoferrin. D. Present in a promyelocyte stage only. So the correct answer is A. Coated with a phospholipid membrane. So why B is not incorrect? Eosinophilic is the primary granules, not specific granules. Specific granule is the secondary granules. The statement is designed to be tricky. Primary granules are called eosinophilic granules, but secondary granules are called specific granules. Why C is incorrect? The primary granules contain myelopulsitis, but lactoferrin. Lactoferrin is a key component of secondary specific granules. And why D is incorrect? Primary granules are produced during a promyelocyte stage, but they are also present in subsequent stages, myelocytes, metamyelocyte, and segmented neutrophil. This statement mentioned promyelocyte stage only, so D is incorrect answer. So move to question number 2. Which of the following are indicators of neutrophilic response to tissue damage or inflammatory stimuli? Correct answer is A. Toxic granules and Dolly-spory in the neutrophils. The combination of toxic granule, it is darker, more prominent primary granules, and Dolly-spories, they are pale blue patches of red endoplasmic reticulum. And vacuolation is collectively known as the toxic granules. These are the classic signs seen on the peripheral smear that indicate a severe inflammatory response, such as bacterial infection and sepsis. Why B is incorrect? Vacuoles are a toxic chain, but the barbories is the inactivated X chromosome. It can be seen as a small drumstick appendage on the nucleus of the neutrophil in females. So this is the normal finding, not an inflammatory indicator. And C. Hypersegmented neutrophils are the characteristics of megaloblastic anemia, B12, or folate deficiency. And orals are abnormal, fused primary granules found in myeloblasts and are diagnosis for acute myeloid leukemia. For D is incorrect because pyknotic neutrophils are dying cells and can be seen in subcells, but they are not specific response indicator. And Russell Boris agar globular inclusion or immunoglobulins found in plasma cells, not neutrophil. So move to next question. What is the time of cell movement through blood vessels to the tissue size? A. Diabetes. B. Opsonization. C. Margination. D. Chemotaxis. Correct answer is A. Diabetes is the specific term for the process where a leukocyte actively squeezes between the endothelial cells that line a blood vessel's wall to access the bloodstream and enter the surrounding tissue. So opsonization, it is the process of coating a pathogen with proteins like antibodies or complement to make it more easily recognized and engaged by phagocytes. So margination is the process where leukocytes slow down and adhere to the inner wall of the blood vessels before they begin to cells. So it is the step before diabetes cells. Chemotaxis is the direct movement of the cell through tissue toward the source of the chemical signals like cytokines from an infection. This occurs after the cell has already left the blood vessel via diabetes. So this slide gives us a perfect visual for the previous question. Here you can see the key events. First, margination. So neutrophil roll around and stick the blood vessel wall. Next, diabetes cells occurs, which is the action of squeezing through the endothelial cells. Finally, once in the tissue space called the chemotaxis to follow the chemical signals, the chemotaxis fed us to precise location of the problem. This diagram helps you distinguish between these closely related pathologic steps. Number 4 is vasodilation and bronchoconstrictions are the results of degranulation by which of the following blood cells: A. Eosinophils, B. monocytes, C. neutrophils, D. basophils. Or A, B, C are incorrect. Why eosinophils, monocytes, and neutrophils are involved in inflammation. They are not the primary source of the histamine that cause the specific side effect. The main roles are related to parasite defense. Eosinophil is parasite defense, and neutrophil and monocytes are phagocytosis. So correct answer is the basophils. Basophils, the large dead granules of the basophils are filled with histamine and heparin. So this slide reinforces the point perfectly. It shows the central roles of histamine in the body's response. You can clearly see that the histamine leads to blood vessels to dilate, that is vasodilation, and bronchoconstriction. Basophils are the circulating cells responsible for releasing these powerful chemicals. Our final question is, how can we tell B and T lymphocyte apart? On what basis can B and T lymphocytes be distinguished? Correct answer is B. Monoclonal antibody reaction to surface and cytoplasmic antigens. This describes the technique of immunotyping most commonly performed by flow cytometry. B cell and T cell have a unique proteins on their cell surface known as clusters of differentiation CD markers. For example, all T cells express CD3 while B cells express CD19 and CD20. Specifically designed monoclonal antibodies can bind to these unique markers, allowing a machine to identify the count the different lymphocyte population precisely. This diagram illustrates the functional differences that are reflected by the surface marker we just talked about. While they look the same, their jobs are very different. T cells like CD8 plus cytotoxic T cell and CD4 plus helper T cells are involved in cell-mediated immunity. B cells differentiate into plasma cells, which are body's factories for making antibodies. We identify and count these different populations using those specific monoclonal antibodies. That concludes our review for today. Remember, the key to ASCPi exam is not just memorizing facts, but understanding the connection between cell morphology, cell function, and disease states. Keep practicing and pay close attention to the details that differentiate similar looking cells or conditions.